we only had to do 2 parts last time, okay? The first of which are always nice and big and obvious, are the seminiferous tubules. Okay. And sort of stuck in between, you can see these sort of little triangular shaped cells. This one's still kind of not that separated, but this is a good one right here. These are the interstitial cells of Leydig. There's kind of another one here. And here's a nice one right here. So we have to know those two, okay, the two basics, the seminiferous tubules and the interstitial cells. But within the seminiferous tubules, we have to get a little bit more specific. So let me find a good one to demonstrate. All right, so here's some good ones. So you can see that some of them are pretty much clear on the inside, okay? But for example, like this one is sort of fuzzy on the inside. I'm going to show you what the difference is here. So with these seminiferous tubules, within them you have, sperm. Well, I'm using this term loosely, but sperm that are in different developmental states. Okay, so some of them are empty because they do not have spermatids in them. Some of them look kind of cloudy because they do have spermatids inside. So let me zoom in first. So with all the seminiferous tubules, whether they have spermatids in them or not, so here would be one that really doesn't have many spermatids in it. Okay, where up here these ones do have spermatids in them. You guys can tell the difference. This one does have them. This one doesn't. I'll show you what they are in a second. Okay? But whether they have spermatids in them or not, you can see making up the seminiferous tubule, these dark, sort of black spots, those are the spermatogonia. And the spermatogonia are what are going to divide to produce the spermatids. So they're basically stem cells. So, well, those are actually more out to the correct. But, so, spermatogonia here. And then here, on this one, you don't really have that many, spermatids. Okay. But in this one, you can see this sort of darker, sort of stringy material, kind of just around the inside. These are the spermatids. Okay, so basically they're the immature sperm. They'll go to the epididymis to mature into full-grown sperm. So this one no spermatids, but this one has spermatids. Sometimes if you look closer, if you get the focus just right, you can see the little heads of them. But what you're mostly seeing is, just the whole thing, which would be the tail with the little head. So spermatogonia, closer toward the outside, these large black dots. Then as you move toward the inside, some of them will have the spermatids, which is sort of this dark stringy material. So with this slide, if I'm being specific and wanting you to answer spermatids or spermatogonia, I kind of have to zoom into at least this magnification and say put it point it here, or go where the spermatids, kind of point it in this cloudy stuff. So I'm less likely to ask you seminiferous tubule on this one because we're learning these two new parts that make it up. Okay, so I'm more likely to ask you spermatids or spermatogonia than seminiferous tubules. Interstitial cells are still up for grabs though. Okay, I could probably label those too. So now another one that we've seen previously, and this is the ovary. So we looked at this also in the endocrine system. So we had to know 2 parts last time, and we're adding additional parts this time. Get everything up. Okay. So one of the parts we had to know last time makes up the bulk of the ovary. It's this sort of grainy purple stuff in the middle. That's the corpus luteum. So all of this stuff, I'm kind of outlining it here. That is all corpus luteum. If you remember last time, the other part we had to know was the Graphian follicle. And we can see one of those here. But this time we're not just doing the Graphian follicle. We also have what are called the primary and secondary follicles. So I'll actually start with the primary and secondary follicles and move up to the graphene. So the primary follicles are found sort of near the edge of the ovary, and they show up as sort of these white little spots. And they're very small. It's really hard for me to get the pointer on. But you can see here, here, here. There's a couple of them here too. Now as they grow and they mature, they become secondary follicles, which you can see sort of around here, okay, which are smaller, smaller in size than the graphene, but larger than, of course, those tiny little primaries. And they'll actually sort of start getting this little clear area on the outside, like you see in the graphene. But the thing with the secondary is that there's no it's not like they're primary follicles and then pop one day they're secondary follicle. Okay, so sometimes you get these sort of intermediate phases where it's a little bit hard to tell, is it primary, is it secondary, is it graphene? But when I do it, I'll pick ones that are really clearly not a primary, right? Because primaries are tiny. Clearly not a graphene, because graphenes are huge. Okay? And then also with graphenes, what makes them stand out? They have a kind of a thick, clear area around the outside, and then you can see clearly defined here. This is a oocyte. Okay? So think of it like a fried egg. Okay? You have the white of the egg and then the yolk of the egg. You'll always see those with the graphene follicle. This one actually has my favorite graphene follicle. I like that one. And you know why I like that one? It has two yolks, right? Two ova. Or I think these are secondary oocytes, I think actually at this phase. Okay. But if these both were released during ovulation and they were both fertilized, it'd be twins, right? So that is it for the